Hey, okay, thank you so much for all the feedback on the other videos that I did. I'm really glad that it helped. So I'm gonna go through chapter 12 and 13 real quick, and I noticed that the revised study guide left a lot of things out and just hit the highlights. So this doesn't seem to be like it's gonna be a lot of material. So you kind of got lucky on that. Okay, so I'm going to go through the brain and spinal cord. Here's what I've got: that revised study guide that we used last time, and the textbook. And I don't even need a whiteboard because everything that we're going to look at is right out of the book. Okay, so it shouldn't take very long. Thank you to my husband who holds the phone. Okay. So the first part on this study guide is it goes through the brain. It just says know the major parts, what are the functions of these regions, where do we perceive certain things. A lot of this is going to overlap between lecture and lab, so this is going to be very advantageous to you. Just a little bit about the anatomy of the brain. I'm over on page 427. This is the external view of of the brain, and you'll notice that it's highly folded in this major part of the brain, which is called the cerebrum, and the cerebellum is right here, and it's folded as well, but it has smaller folds, and then the brainstem's going to come off the bottom. I want to talk about this just for a minute. You'll notice how it's divided up into lobes named for the cranial bone that is directly on top of that lobe that's protecting that part of the brain. So we've got frontal, parietal, temporal, and occipital lobes. But then you'll also notice that it's highly folded. So what are these folds called? Well, the shallow depressions, or the grooves, are called sulci, or your book says sulci, and I've never heard it said that way, but whatever. It's SULCI. And so those are the little depressions that we see. So those depressions, that's actually the central sulcus. It's a major depression that separates the frontal and parietal lobes. And but then we have smaller ones. And then the little ridges, the elevated ridges in between the sulci, like a ridge, is called the gyri, or they call it the gyri. Gyrus is singular. And so the convolutions of the brain, which are the folds, they are made up of these sulci and gyri, which actually increase the surface area of the brain so you can have more neurons. And when we talk about how many neurons we have, it's in the billions. So it's extensive. You probably heard that we only use like 10% of our brain, and that's not true at all. We use a lot of our brain. We use all of our brain. The brain is like outer space or the deep oceans. It's a big mystery. There's so many things we don't know about the brain, but anyway, that's sort of the makeup. That's probably what I just went over probably will help you more in lab than lecture. But anyway, the major, parts of the brain, I want to actually use a mid-sagittal view of the brain. This is what you'll probably see mostly in lab. The reason I like to use this diagram, which is on page 435, is because we can see all the parts. We can see the lobes. Okay, so we see the lobes of the cerebrum. Okay, we see the cerebellum, and we notice it also. Well, it comes in a hemisphere. So the cerebrum and the cerebellum both have a right and a left half. And so we call, I'm sure you've heard of like the right brain and the left brain, but we call them the hemispheres. And so we're actually just looking at one half of that. But why I like this image is because we can also see the midbrain, which is composed of the thalamus, the hypothalamus, the epithalamus. And your study guide asks you to talk about the functions of all of these regions, and all that is right in the book. And so that's not anything difficult. It also talks about where do you perceive certain sensory information like smell and taste, vision and hearing. That's also directly in the book. I don't think it's going to be hard to find it all. I just wanted to point out some of these areas. Okay, so this is the cerebrum, and this is called the corpus callosum, and this is actually a region of the brain that connects the left and right hemispheres to each other. And then in the central portion, we call that the diencephalon, but it contains the thalamus, the hypothalamus. And how do you know that's the hypothalamus? Because the pituitary gland is hanging right off of it. So when you see the pituitary gland, go just above it and you know that you're in the hypothalamus. And then just go up just above that and you know that you're in the thalamus. And then there's another important gland in here called the pineal gland. And this is a little endocrine gland actually that produces a hormone called melatonin, not to be confused with melanin, that regulates sleep-wake cycles and seasonal breeding patterns in other animals. And then we have the brain stem. And you notice the brain stem looks like a backwards P in this picture, but that helps us remember that this region's called the pons. It sort of looks like a P, but we have the midbrain, the pons, and the medulla oblongata, which is actually continuous with the spinal cord. So you come out here and it just keeps going down, and that's the spinal cord. So those are the major regions of the brain. I would like to talk about the cerebral cortex for just a minute. The cerebral cortex is the outer few millimeters of the cerebrum, just the outer few millimeters. But so many important things happen in the cerebral cortex. Higher ordered thinking, voluntary commands, motor commands, cognitive that means learning, how we learn, our personality, speech, memories, all of those things happen in the cerebral cortex. So the cerebral cortex is extremely important. If you go back over to page 428, you'll see the structures of the cerebral cortex. Remember, this is just the outer few millimeters of the cerebrum, but it talks about, and here we see the central sulcus is our separating line for the primary motor cortex that is in red, the primary somatosensory cortex, which is in blue. So this is voluntary motor commands, and then this is like the receiving of all those sensory input that's going to come in. There's your primary visual cortex. So there's the answer to one of the questions. That's where our vision happens. It's in the occipital lobe. Auditory association areas. There are two important areas that your book asks about as far as speech. It's Broca's area, which is here, and then the Wernicke area, which I'm not sure where that went. But these have to do with not just can we come up with words, but it's actually like the motor commands to move our mouth to actually produce words. That's what happens in Broca's area, but you can look both of those up, okay? And that's under the brain. Letter E, and then Roman numeral 3 and 4 on the study guide. Okay, the next thing they talk about are the meninges, so we kind of skip a lot. There's the cerebellum. That helps with like coordinated muscle movements. Like if you watch The Walking Dead, you notice how the zombies, like how they walk. That's called ataxia, but because you don't have smooth coordinated muscle movements, but the cerebellum coordinates those movements for you. And especially if you have some type of learned skill like dancing or some type of sport where you do this repeated skill over and over, the cerebellum's responsible for that. They skip a lot of stuff. Okay, protection of the brain. This is over on page 450. So these are the meninges, and I'm sure you've heard of the meninges, and there's 3 layers to the meninges. If you look, I'm over on page 450, but we have the dura mater, which is directly under the skull, and that's the outermost layer of the meninges, and Then we have the arachnoid mater, which is underneath that, and then the pia mater, which is the third layer, and it's directly on the surface of the brain. Not only does this protect the brain, but it also protects the spinal cord. And then circulating throughout a few of these layers, we also have the cerebrospinal fluid, and they ask you about that. Now, the cerebrospinal fluid is formed in what are called the ventricles of the brain. And if you find a weird picture that looks like ram's horns, hold on to 1 second. Probably making your car sick with the move in here. You see this blue, how that looks like these big sheep horns or something and coming around like this? Those are called the lateral ventricles, and then we have a third ventricle and a fourth ventricle that runs down in here. But the ventricles are these big chambers inside the brain where the cerebrospinal fluid is formed. And if you remember back to your neuroglia, they actually help form, one of those cells helps form the cerebrospinal fluid. You can find out which one that is. But anyway, page 451 goes over all of the meninges. Here's a good picture of those ventricles, the ventricles of the brain where the cerebrospinal fluid is located. It has its own circulation system throughout the brain. This is over on page 453, and it shows you exactly how the cerebrospinal fluid circulates. It goes through the cord, around the brain, in the brain, all over. At any given time, you've got about 2/3 of a cup of cerebrospinal fluid just circulating around the brain and spinal cord. It's protective, okay? That's one reason that we have it, but it also contains nutrients, eliminates wastes, anything that anything that fluid in your body would do. It maintains temperature, buoyancy of the brain, which is not something we probably think about much, but a lot of functions there. And I'll just go ahead and remind you, it's the ependymal cells that actually form the cerebrospinal fluid, and they do it by extracting plasma out of blood. Okay, so it's fluid in the body is all the same. It just depends on where it's located. It has a different name, right? So when it's in the brain, it is cerebrospinal fluid. Okay, and then we have the they ask you a question about the blood-brain barrier. There is a little description about the blood-brain barrier, very important, page 454, if you want to know about that. Okay, the spinal cord is next, and it is very important that you are able to label a cross-section of the spinal cord. Okay, so when you look at the spinal cord, so here's the cord. You see the meninges surrounding it because it also protects the cord. When you do this cross-section, we end up with this interesting little butterfly, okay, little image here, but this is because of the gray and white matter that I mentioned before. That's what forms this little H. We call it the gray H because that's the gray matter, and then that's the white matter. And then there's our central canal. That's where the cerebrospinal fluid flows through. But what you notice is coming out of both sides of the cord is your spinal nerve. Okay, we had 31 pair of those spinal nerves. When they come and actually, if they're either one portion's actually leaving the cord and one's coming in. Okay, so that's the motor root and the sensory root, or we call it the ventral root and the dorsal root. So then it fuses out here, and that's your spinal nerve, and that's one of the pair of your spinal nerves. There's probably a better cross-section somewhere like right here over on page 458. And so this gives you a little more. They call these horns. Okay, so how do you know, like, if you're looking at the front or the back of the cord? Okay, look for this big fissure right here. That's called the anterior median fissure, and you're at the front, so that's the front of the cord. And then you've got these horns on the gray H. That's the anterior horns, the posterior horns, the lateral horns. Okay, And notice that over here, our anterior root is motor neurons. So they're leaving the cord, going out to their targets. The posterior root is sensory, so it's coming in and bringing information. And then the type of neuron that we see in sensory neurons, their cell bodies are off to the side. So what happens is it forms this little ganglion, okay, or this little knob, and this is called the dorsal root ganglion. And that's where the cell bodies of these sensory neurons are located, and that's why it's enlarged. So these are very key features on the cross section of the cord. You're going to need to know this for lecture and lab. So, and I think I just went through a bunch of stuff there. Okay, it says, what are tracks? Well, tracks are in the white matter, and the tracks, it's over on page 459, they're like super highways that run up and down the cord to the brain and from the brain. So when we get information from spinal nerves and they come into the cord, they've got to get to the brain. So what do? They get in these tracks. Then the tracks go up to the brain. Then the brain is going to analyze this information and send out motor information back down these tracks. And so they're ascending tracks and they're descending tracks. So you've got some going up to the brain taking information. You've got some coming down from the brain bringing motor information. So what would be going up? Sensory information. What would be coming back down to go back out? Motor information. Okay, so that's important to know. Okay. And it gets complicated, but I think they skipped all that. So, there are some special senses. They talk about, we're going to talk more about that later. The spinal nerves, you just need to know the nerve plexus. I don't even know where that is. Okay, the one thing I did not mention were the cranial nerves. And the cranial nerves, I don't remember where I saw those. Sorry for making you car sick here. I know they're somewhere. Well, I know they're in your lab manual. The cranial nerves, there are 12 paired cranial nerves that exit out from the brainstem. And in your lab manual, there's a whole page on those. They have a Roman numeral, they have a name, and they have a function. And you need to know all of those for lecture and for lab. So that's number 2 there. Know the cranial nerves. Okay. Now, as far as the spinal nerves go, they only list a few right here. Brief description of function and know the nerve plexus it originates from, and that's also that's in your lab manual. I know. We've talked about a reflex arc when we talked about sensory receptors in the skin in lab. We talked about how you hit a sharp tack, it sends a signal to the cord, and then to maintain homeostasis, it sends out a motor signal for your contraction so that your arm moves away from that stimulus. These are some things that, like that was called the withdrawal reflex, and so that was something we've already mentioned. You can go back and look at that. Everything else, I think you shouldn't have any problems finding in the textbook, where they just talk about the differences between like afferent and efferent pathways, and I don't think that's anything difficult. This is getting to be pretty long, so I'm going to stop here. That should cover everything. Remember, I'll open up the test next Monday. I believe that's the date that it opens. It'll be very similar to... let me talk to you to my face. It'll be very similar to the other tests. So, you know what the deal was with that. I'm trying to think of anything else, but I can't really. If you have an issue, the discussion forum is a good place to ask questions. I like that because, somebody else may have the same question you have. And then everybody can read the replies. Thanks to everybody who said the lecture videos were helpful. I don't mind doing them at all. We're about to wind it up. So this is test 5. We only have one more. And goodness, we'll be getting toward the end. So if you need anything, just email me. I hope everybody's well and safe. Stay safe. Don't go anywhere. And I will talk to you later. Bye.